This is Stephanie Langel. I am currently now on Dairy Carrie's Facebook page. I think before I had gone live on my own Facebook page, so that was my fault. And then we had a couple issues with actually getting a really good connection. So let me just double check to make sure that I'm on the right page. I on your page. And I think one of the things that I was discussing before was the fact that everybody's working virtually right now. And so I'm on Duke Internet and this should work just fine. But I think because a lot of people are signing into the VPN or the virtual private network, it's kind of overloaded. So maybe that's why I was having issues before. But it seems like Carrie's giving me the thumbs up that it's working for me live on her page. So I'm going to check in with everyone. Ugh. Every time I click onto Facebook, my face comes back up. I'm trying to create a system where I have something over my face and then your wonderful comments are on the right-hand side. So I'm seeing people are joining, which is great. And I'm going to give everyone a couple minutes to kind of join and get in, and I will stay in touch with Carrie to make sure that everything is working properly. It's my first time doing a Facebook Live, so hopefully you're able to forgive me for any issues I've had starting this up. So I can introduce myself first. So as I mentioned, my name is Stephanie Langel. I am a postdoctoral scholar at the Duke Human Vaccine Institute. And I came from Ohio State. So I moved down south. It's a bit warmer here. It's really nice in the Raleigh-Durham area. We're really enjoying it. Of course, we do miss living in Ohio. And I think the Buckeye State will always be home for me. It's funny, my husband and I are very much Midwesterners because down here when it gets even into the 60s, people are wearing coats and they're wearing gloves and we just think it's hilarious. So we hope we don't become too accustomed to this weather and lose our Midwesternness. But we really do enjoy it. And I came from doing a PhD actually on a coronavirus. And the coronavirus that I did my PhD on was a porcine coronavirus. So I was in the veterinary school and focused on particularly animal coronaviruses. One thing we'll talk about is how coronaviruses are really good at jumping species from animal to animal. And we're animals, so they jump into humans as well. They jump back out of humans into other animals. And it's just the way that this virus, the machinery of this virus that makes that be the case. So in 2013, if you were a pork producer or somebody who was involved in that part of the food chain, you probably heard of porcine epidemic diarrhea virus, which was a virus that we've known about for a long time, but it mostly was in Europe and then in Asian countries, but it came in 2013 to the US and wiped out 10% of the pig population, mostly impacting neonatal piglets. It could cause up, if you got it in your herd, you could cause up to 90 to 100% mortality of those baby pigs, which, as you can imagine, as a farmer, it would just be devastating. And so that's an example of a coronavirus that came to a immunologically naive population. None of the pigs in this country had ever seen that virus, and so nobody was protected. And so that's why it really, once it got onto a farm, even with the high biosecurity standards that pork farmers have in this country, it really could wipe out an entire herd and was a really big problem. And based on that experience and experiences that we've had in the past with veterinary coronaviruses, we've known for a long time about their epidemic potential. They have an ability to replicate in different body parts depending on what species or what type of coronavirus it is. Some of the farmers who work with cattle are probably familiar with the bovine coronavirus. That's a respiratory virus. The virus I mentioned before, the porcine epidemic diarrhea virus, obviously isn't enteric. It's in the gut. And so both of those cause a lot of problems, particularly for the bovine coronaviruses during stress, times of shipping, can cause issues. And so that really kind of set up my background for understanding coronaviruses, because what I studied was building immunity to them. So how would a completely naive population, which is what the swine population in the United States was when PEDV first was introduced to the United States, how do you build immunity in a population of naive, I'll say individuals, but of a naive animals? And so after my PhD, I had wrapped that up in 2018. I did my PhD just to with Linda Safe at Ohio State. I had excellent mentors in coronavirology. Anastasia Vlasova was another one, and Shuhong Wang. And I just was really well set up to then move on to the next stage. And if you're not familiar with kind of how an academic research trajectory works, typically, if you want to stay in academic research, you do a postdoc. And so that's where I'm at now at Duke. Duke has something called a Human Vaccine Institute, and we study viruses of all different kinds. Particularly in my lab, we study viruses that transmit through the placenta, that can transmit through breast milk, and that impact the mom and the baby. So some of these viruses are HIV, CMV, which is called cytomegalovirus. Some of the others we work with Zika. And the virus that I work on is called rotavirus, which does not pass through placenta or through breast milk, but does really can devastate young children, particularly in third world countries. So we're focused on trying to identify ways to protect these vulnerable populations in our lab. And what we're now doing also is working on the new coronavirus. So that is a little bit about me and my background and where I came from as an animal coronavirologist or animal coronavirus immunologist, is trying to understand how these viruses work and why particularly they can pose problems in a really naive population being as infectious as this one is. So that's a little bit of an introduction, and hopefully we can, getting some fun comments. Yes, I'm glad some people found me over here. Sorry, I posted on my Facebook at first, but now I'm on Derek Carey's. So Carey had sent me some really excellent questions, but you can ask any questions you may have as I work through some of these to try to give a better understanding of this virus. So this virus has been named. It's called SARS-CoV-2. And the reason that is there is a committee called the International Committee for the Taxonomy of Viruses, and they meet whenever we have a brand new virus to name. And what they do is they consider a lot of things. They consider the types of presentation of the symptoms. They consider the actual sequence of the virus, or the genetic parts of that virus, and they try to find, okay, where does this virus fit within all the other viruses that we have identified? And this virus causes the same symptoms as SARS-1. So if this is SARS-2, what SARS-1 refers to is a virus that emerged in China in 2002. That SARS-1 lasted 1 year. There were a couple cases in 2004, but pretty much lasted a year. It killed about 8,000, or infected about 8,000, infected 8,000, killed about 700, and it was gone after that. And we did learn a lot from SARS-1, but we really, unfortunately, did not learn enough to what we would have liked to based on this current pandemic. And it goes back to how science works in this country, unfortunately. I mean, when you're trying to convince, so we're mostly funded through governmental bodies, through taxpayer dollars. So thank you to all of you for funding my PhD through USDA, but then also my current work through the National Institutes of Health, or NIH. I mean, really the way I perceive science is doing this for you and doing this for the public. And it really is, for me, motivating and something that kind of gets me up every day and gets me excited to do my work. Is not only am I discovering potentially brand new things about these viruses, but I'm doing it for you. So, but what that also means is you're trying to convince the public and Congress to care about a virus that showed up in China for a year. It killed like 700 people. Again, a second emerging coronavirus came in 2012 in the Kingdom of Saudi Arabia, and that was called MERS, or Middle Eastern Respiratory Syndrome. It's a coronavirus. It has similar tropisms. And when I say tropisms, I just mean its ability to infect a certain body part, so in the lungs, causing respiratory distress syndrome. And it came also from an animal reservoir, which we'll talk about in a little bit. But after SARS-1, it blipped for a year, and then MERS came, and MERS has stuck around. It's been hard to completely eradicate that virus. But with it going away, never coming to the United States, what would be the motivation to fund, okay, let's find antivirals that work for every coronavirus out there. Let's try to develop a universal coronavirus vaccine. Of course, these are things that scientists have been arguing in the coronavirology field for years. We've known a lot about these viruses and the pandemic potential they have, but the problem is if you have influenza, which kills hundreds of thousands of people over the course of time, if you have HIV that devastates and can integrate into your genome, and we have drugs, but you can't get rid of it, you have a lot of other things you have to worry about in terms of public health. And there's only a certain pot of money, which, of course, scientists, we would argue, is too low. And recently, unfortunately, a lot of the pandemic and epidemic surveillance networks have been shut down from funding cuts. So we would argue that we just weren't able to work on coronaviruses the way that we would like to, because the funding, now there's an $8 billion funding package for this, but it's because it's here and it's a problem. So it really is just the way science is funded, and that's like a whole another conversation, which I'm glad I'm not a policymaker, but I'm an advocate for supporting your local or state, national governments to try to fund NIH, USDA, but that's a whole nother conversation. So what I just described then was two emerging coronaviruses, SARS and MERS, and now we have the third, which is SARS-CoV-2. That's the virus name based on science and based on the ICTV, International Committee of Taxonomy of Viruses. They meet, and it's very special, and they get to name the viruses. But the disease name was named by the WHO, and that's COVID-19. I'm not a huge fan of that name. I think, okay, we have another one that's like COVID-29 if it comes in 2029. But regardless, that's the disease name because it causes really the same type of respiratory distress symptoms that previous ones have. So what we know about this virus and kind of when it emerged. So I'll kind of work my way into that. And I am seeing some questions that I'm going to keep in mind. I think somebody named Dwayne asked about shipping fever in cattle. Those are some good questions. I'll kind of try to circle back from that. Oh, hi from LA, sunny but feeling isolated. That was Mimi. Yeah, I know it can be tough on mental health right now. The way I'm going to progress through this talk is I've introduced myself, talked about my background, introduced the previous two coronavirus outbreaks, and now I'm going to start on the current one, but I'm going to eventually get to like the public view, the consequences that are happening right now. So I'll try to work my way to the end there. But this virus, we knew as an international community on December 31st, when the WHO announced that there was an outbreak, several cases of pneumonia with unknown etiology, and etiology just being from unknown causes, were reported in Wuhan, China, to the WHO. So December 31st is when that happened, and really then over the course of January, you started to see numbers of infected people increase, and then death as well. And those numbers have been reported. There's a really excellent website by Johns Hopkins that I can share with Carrie's page that really is an ongoing surveillance of the numbers of people who've been infected in each country. So if you've kind of been wondering, where do I find these numbers? Johns Hopkins is doing a really excellent job of keeping that together. And so after December 31st, when we started seeing cases in the international community, got quite worried about this because of the rapid rate of spread in Wuhan, China. And they were able to quickly determine it was a coronavirus because of the techniques we have in molecular biology. So we're able to tell that it's a coronavirus based on its genome sequence and you can look at similarities amongst its genome and say, okay, that's a coronavirus because all these coronaviruses have those parts inside of it. And then you can start categorizing this coronavirus. Okay, it's not this in the same genus as my porcine coronavirus that I studied, but it actually is in the same genus as the bovine coronavirus, but in a different subgroup. And so that's where you start kind of separating out what types of viruses these are, and those have big implications for how they infect its host species or humans. And so after they were able, so what they were doing is they were finding people with viral pneumonia, but these people were negative for flu, because you usually at a hospital, there's a panel of viruses when you present with pneumonia that you can try to detect for. And then once they're negative for flu and other things that would cause viral pneumonia, you then look further to try to say, well, what could this be? And because we know that coronaviruses have caused problems in the past, it's not completely surprising that it might be. So they were then able to use something called PCR, or polymerase chain reaction, which is just a way to break down the genome of the virus and detect certain parts of it that say, oh yes, that is a coronavirus. And so after then they isolate the virus from the clinical specimens in the lungs, they then brought it back to the lab, and you can take cells that are in culture and you infect them, and then you can say, okay, yes, this is a virus, it's a coronavirus, it causes infection in cells, and let's look at it under a microscope. And it has this very characteristic shape, which you've probably heard talked about, is why it's named corona, because it's Latin for crown. And it has very particular, and I have like a really nice picture on my phone. I was wondering if I'm going to zoom in and see if I can show this to you. I was going to do a screen share, but I don't know, my computer wasn't really being nice to me. So what I'm showing you right here, let's see if I can get the glare out of the way. That's what a schematic of a coronavirus looks like. And what you're looking at here on the outside is that crown. And those are called spike proteins. So you can see it's actually pointing, it says S. It just is a spike protein. So that's what gives it its characteristic shape. And that's what it looks like under the microscope in the respiratory tract of infected individuals. And those spike proteins on the outside that create the crown are really important for how this virus can infect our lungs, but specifically certain receptors or proteins on our lungs that it grabs a hold of and then can start replicating in the cells of our lungs, causing viral pneumonia, killing cells in our lung. And then basically, well, there's a couple things that viral pneumonia in terms of symptoms can cause, but it ruins the lining of the cells, and then fluid cannot escape out, and basically you kind of drown in your own lungs because you can't, the fluid, just the buildup, and you're not able to control that anymore because the epithelial lining has been wiped out by the virus. So after we really started to see such a huge amount of spread in January, public health officials and scientists, we were monitoring this really since it broke in WHO. And we were concerned because the numbers increased at a rate that was, very different from, well, it was similar to the first SARS and different from some other viruses. So it really alerted people to think, okay, this could be a problem. And I think maybe some people saw in the news as well that they were building hospitals in that area where the original infection happened quite quickly. I think rapid 10 days of hospital for thousands of people. And actually that was when I just had a feeling that this was going to be quite bad. I don't think you build hospitals at breakneck speed for thousands and thousands of people if you don't have an infection that spreads quite quickly. That's overwhelming the hospital system in that area. So what kind of this leads to is I'm going to bring up some of Carrie's questions that she posed to me that some people have been asking, because it really, a lot of this leads to the question of where did it come from? And a lot of these viruses, actually coronaviruses, they come from bats. And we know this because people have been sampling bats for the past decade or so. There's a couple really excellent researchers, both in the US and in China, who they go out and they collect bat guano, and guano is the feces of bats. And what they can do is they can take that back to the lab and they can isolate the viruses that are in that guano. And really for the past 10 years or so, coronavirologists have been reporting, and I wonder if I could pull up the titles of the papers that have been published from those experiments in like 2015. Let me see here. Yeah, I have it right here. I'm just going to read it for you. So here's the titles of some papers that the scientific community, we've been discussing for a while. A SARS-like cluster of circulating bat coronaviruses shows potential for human emergence. That was published in 2015. SARS-like coronavirus poised for human emergence. That was also published, that was in 2016. And then further evidence that bats are the evolutionary source for MERS, or Middle Eastern respiratory coronavirus. So because we've been sampling bats and looking at their guano, we've known that there are viruses in bats, circulating in bats naturally, that look very similar and have similar characteristics to viruses that infect humans, which would be SARS-1. SARS-1 would be the reference virus for that. And what's really amazing about coronaviruses in bats is, one, they replicate, they are in the gut. They're not in the respiratory tract, at least that we know of, compared to what we're being infected with right now. But the bats are completely healthy. They're fine. They fly around. They crap. There are guanos everywhere. And in that guano are viruses that are poised for human emergence. Poised being they have a potential, if ingested by us or if in contact with our mucosal membranes, to bind to our receptors that then allow them to enter our body and replicate and promote disease. So these are things that were known, and how we know it came from a bat is because we're able to sequence the virus. If you work in any type of genetics within agriculture, you know the technology is such that we can sequence an entire genome and look at what's, what the contents of that genome are, so that we're able to, determine, okay, the genome looks like this. Like, I have a whiteboard behind me, so let me see if I can show you. So if the genome looks like this, and it has, let me see if you can see that. Yeah, my big old head's in the way. There we go. So if the genome looks like that, and there are certain types of mutations in that genome, certain characteristics that are represented by the blue, I'm just going to draw. I mean, it's much more complicated than that, but we don't really need to make it complicated. This is simple enough. If it looks like that in a bat, and then you sequence this new SARS virus, and it's very, very close. It has this gene, this, but not this. We can say, this is 98 to 99% similar. Then we know with a high factor of confidence that they're related. Those two viruses are related. So over the course of time, this original bat virus has developed mutations that made it able to then jump into humans. So let's put that down here. Humans is this one. And when I say mutate, it sounds scary, and it's something that the media talks about a lot, but viruses mutate all the time. It's what they do. They have very small genomes. So our genome that makes up our body is huge. Viruses, it's very small. And to even get on a tangent about viruses, I mean, they're amazing because they're not alive. I talk about them a lot as if they're alive because anthropomorphizing viruses makes them easier to understand, but they have no goal other than they are self-assembled proteins that able to hijack your body to replicate themselves, which is amazing. I mean, the fact that they're not living and they're doing this and they're causing all this destruction is why viruses will always fascinate me and why I love being in this field. But they're going to mutate over the course of time inside the bats or inside whatever host species they're in. And as they develop mutations, those mutations could lead to nothing. They could be detrimental to the virus. They could stop the virus from replicating, or those mutations could allow a very small change in its ability to bind to the lung cells inside its host. So let's say it changes in just a way randomly, because all of this is random. The mutations are random, evolution is random. It changes in just a way that it binds to human lungs with an affinity or with a strength that's just a little bit greater than what it was before. And that's how you can start to see how these viruses in particular and the way they replicate, which is more, I mean, people are interested in learning more about this, we can talk more in like a more molecular depth, but really that's what makes coronaviruses and their ability to jump species is that they're replicating in a way along host species that if they change a little bit and they're able to maybe more tightly bind to our lung epithelial cells, and then we somehow come in contact with these animals, which is key, then maybe we pick up that virus and randomly, because of a mutation or something in the virus, it starts replicating in us and causing disease in us. So that's kind of how to think about these viruses. It's not a process that's necessarily surprising or just like happens. It's very slow over time that these mutations happen, but one mutation could be favorable to jumping species. And so how then do you come in contact with animals, and how could you pick up this virus in bat guano? There was a lot of conversation about eating bats, and that goes back to originally this virus was thought, a lot of it, when you do epidemiology and you're trying to understand where a virus came from, you're asking people, okay, where were you when you started feeling sick? And where a lot of people were was the seafood market. Not particularly surprising. I mean, a lot of people go to the market, they have to eat, they want to get food, but it was, a commonality amongst the people who were getting sick was that they were at the Wuhan seafood market. And so that kind of led people to think, okay, well, markets are a place where there's a lot of interaction with a lot of different types of animals. So this is a place where farmers from the rural parts of China who farm many different animals will bring in animals, live animals. And that is the difference here. I mean, they're not already, well, a lot of them are already killed, but there's a lot of live animals that come in because like you buy live lobster, some people just want something that's fresh. They want something that's live and fresh, they kill themselves and then they can go home. And so it's always the possibility that someone, had killed a bat and brought it for people to eat. But the other thing is that, when you're a farmer living, well, I mean, there's a lot of farmers that are listening to this probably. I mean, you're in rural areas and there are a lot of bat colonies in rural China. I mean, if a bat poops on you, or if a bat poops on your field, or if a bat, remember these viruses are in guano, they're in bat poop. So it is not surprising to think that somebody who sells things at this live market, who came from a rural area that has these certain species of bats, there's horseshoe bats, there's all kinds of different kinds, that have these viruses could make it to the market. So we don't really know exactly how. And so, while shutting down live animal markets might sound, appealing to some, because I know that there's a lot of animal welfare concerns there, it's not going to stop these viruses from interacting with humans and potentially infecting and causing a pandemic, because people have to eat, people have to farm, and they farm in rural areas where bats are. In addition, as our populations grow, we start to encroach upon animal species and disrupt their environment, which means we're in more contact with them. So what's interesting about this virus is while we know it came from bats, we don't know if there is a species. So if this is a bat and here are humans, what we don't really know is there a species in between bat and humans that this virus has jumped between. For SARS-1, there were animals that were in between bats and humans. They're called civets or some type of coon. I think it's called coon dogs, and they're like these raccoon-looking things that they farm and eat for sustenance, for meat. And so the virus had patches through that. And then in MERS, they're camels, and so camels were getting respiratory infections, and the camel farmers or camel handlers for racing industries, they were getting sick from that. So that hopefully answers your first question, is where this comes from. I don't know if if it came from somebody eating a bat. It's probably more likely because that's not as common, just eating bats, as eating just like other things that you would normally think of, like they're regular farmed animals. But it's possible. It's hard to say, but it definitely came from a bat originally. And so after that, then I think one of the things that's interesting to talk about is, I'm looking at Carrie's questions. Is there a chance this virus was man-made and was released from a lab? So that's definitely, a question that scientists had. I think when something like this happens, when a virus emerges and you don't know where exactly it came from, and you're doing the genetic sequencing of the virus, and you're trying to understand, okay, what is it related to? If it's related to this and this, then we know better, what animal it comes from. But there's always going to be that question, was this virus man-made? So, what you do with that question is you remove your biases and you ask the hypothesis, or you ask the question, does it come from a lab? So the hypothesis would be, this virus came from a lab, or this virus did not come from a lab. And you test that hypothesis based on the genetic tools that we have, based on the sequences that we have stockpiled from discovering previous coronaviruses, from genetic analysis and computer simulations, where we can actually take the genetic information of these previous viruses and use them to model predictions for how certain viruses will bind to the receptors in us and animals. So you take all of these tools, which have really progressed in the last 10 years. It's kind of an amazing time to be a scientist, because you have so many tools at your disposal to try to figure out these questions. And so what you do then is you test that hypothesis, and you try to see, okay, we have the same tools in the US. Could we make this virus? Is this something that we could do? Is it probable that SARS-CoV-2 emerged through a laboratory? And so one of the reasons that one of the pieces of evidence, and I like to talk about things through evidence. I think and work through data, not by rumors or conspiracy. Whatever the data shows me is, then let's pursue that. And so when you look at the sequence, when you look at the SARS-2 sequence and its genome, what's different about it is it has a particular spot called a receptor binding domain. This receptor binding domain is actually really important in binding to the host cells, so our lung epithelial cells. And what was really interesting about this one particular domain is that what we know about coronaviruses said that if this certain sequence was in this virus, it's going to bind less well to human epithelial cells. And so when you look at the sequence, and that sequence is in there, is in SARS-2, if somebody were to make this virus, they would never put a sequence that would cause less binding to human receptors. And that's one of the things about this virus is it's not genetically optimized to be really good at binding the receptor in our cells. And so that's kind of a clue. It's a clue to say, if this was man-made, if I made this virus, I would never choose that genetic sequence. Like, that would be dumb, because it doesn't bind well to the epithelial cells. I need a virus that's going to bind well and potentiate and be able to transmit through populations. And so that's one reason. And then one of the other things is, when we build, like, if you're going to build a virus, you don't just build it like a factory builds a car. You're going to build it more like if you have a Honda Civic and you're swapping out the engine from a different manufacturer, or swapping out the wheels. So with a virus, we have a backbone, and then on that backbone, a new virus is built. You could put the pieces on. But if you have our new virus, you would be able to identify the backbone. And what we know is there is no known backbones of this virus globally. There's nothing published. There's nothing that can suggest that this is something that somebody could come up with. And so it's really hard for us to imagine that a specific group in China was creating a brand new backbone to this virus. And the reason we say that is because we have the same tools here. We have people who are equally as smart. And this is something that we could not do. really this is something, nature is much better at the ability of viruses to mutate and adapt to the host. So it's kind of another piece. Now there's some other genetic pieces that I won't get into, that just the evidence for a lab-made virus really just isn't there. And so that's what we go with. I think we go with the data that says that this is something that over years and decades that has evolutionarily been driven in bats, maybe some other animal species, then to adapt to humans. So hopefully that answers the questions. It can be very technical, and so if anybody ever wants to talk more about that, we surely can. I'm going to jump to Carrie has a couple other questions. Lots of people are reporting that they had an illness with the same symptoms earlier this winter. Is that possible? That this virus has been here for months? And yes, in fact, I would say it absolutely has been here for months. And it's possible that the people reporting those symptoms earlier in the year was from this virus. There's an excellent group at University of Washington in Seattle who, you know, maybe have heard that kind of the epicenter of this is Seattle. And they've been tracking this virus and really have demonstrated through genetic sequencing and looking at how the virus is mutated over time, that it's been here probably since January, maybe even before. I don't think people realize how many planes from large companies like Google and anything out on the West Coast would go in and out of Wuhan on a daily basis. It was an economic hub. It's a place where people go. And so the restrictions on travel probably, it was not going to be able to stop the spread, because what we now know as we look back at historical samples in China, it was in China maybe in November. And so because of that, it has been circulating. And really the inability to test if we have it is what's hindering us to know these questions. And I think that's one thing that you hear scientists and public health officials talk about, is that we don't have the testing capacity right now to know how many people are infected. We didn't have the test in the very beginning, and everything sort of didn't get off to a good start. Typically, for instance, with the H1N1 pandemic in 2009, it was declared a national emergency 2 days later. With this one, it was 11 days later. And that's a critical amount of time to mobilize the amount of testing that we would need for a virus that's as infectious as this one is. So, I think talking about what should have happened, it really is irrelevant to now, but what needs to happen is a huge ramping up of our country's ability to test, which is why it can be hard to understand what a mortality rate for this virus is, because we don't know the denominator, right? So the mortality is how many people died. And if I am incorrect in any of this, I have epidemiologists, you can yell at me if I'm incorrect, but how many people died over how many people were infected. The problem with that is we don't know this number. And that's because we're not testing. I think the last estimate was 5 to 10% of our population can be tested, but this virus is spreading faster than that. The reproduction rate, which is called R naught, it doesn't really matter what it's called, but basically, it's how many people are not R0. How many people can one infected person contract this virus to, and that's what that is. And it's showing by the data that this virus has a higher R naught than flu. So potentially between 2 to 3 people can be infected by one person. And what's been really interesting is there are people called super spreaders, which seems to be a specific type of infected person for coronaviruses. You don't usually see super spreaders a lot, maybe in influenza, but there were some reports that one infected person walking through a hospital could infect, up to 15 people just within the time that they were there. And they might not always show symptoms. And so that's kind of the second thing about this virus that makes it very, very difficult to get a hold of, is it's taking longer for this virus to show symptoms than influenza. So 4 to 7 days is kind of the average. For flu, it's earlier. For SARS-1, it was shorter. It was 24 to 48 hours you would see symptoms. And so you can imagine public health officials and doctors' ability to control a virus that takes 4 to 7 days to show symptoms, but you are shedding the virus during that time. It's harder to control that because you can't get people care, you can't get people quarantined or in the hospital. Then if you start showing symptoms right away, well, you're in bed, you're sick, you're not feeling good, you're going to the hospital. It's a lot harder, and I think that's why we're seeing the amount of spread that we're seeing, particularly in the countries that did not do a good job of being ahead of the game in terms of restrictions and testing. It's very hard to stop once that is going, because there's a lot of asymptomatic spread is what it's called, when you are spreading the virus but you're not showing symptoms. And so it's concerning because younger people who maybe aren't as affected are spreading it, children even, but they don't have symptoms, and so it can be hard to track that. So that question that Carrie posed was a great one because, yeah, it's probably been here for a while, and really our ability to understand where we go from here, how long do we have to, keep our economy on lockdown, is really going to depend on our ability to know how many people are infected and then how many people become immune, because people will develop immunity to this virus. That's another question that Carrie has. And actually, I'm going to check in on people. I haven't scrolled. Oh, I'm glad people are liking this. Great. Okay, good. When do you predict it will peak in the US? That is a great question. We don't really know, but right, predict is the key word. What we know from SARS-1, is that it peaked, if I remember correctly, it peaked in late spring, April, May, and then kind of trickled into June and July. And so I think when a lot of people are talking about what can we expect, they're looking at past experiences with SARS-1. And they're looking at, what happened with SARS-1 and when it was stopped. The problem with SARS, comparing to SARS-1, is that, as I mentioned before, you felt symptoms much quicker. So the strategy in China, once they figured out the virus and what was causing it and why people were getting sick, you get them in a hospital. Because if they're in a hospital and you can control community spread, you might have heard about community spread a lot in the news. Community spread is just, it's very hard to control once it happens. If everything is in a hospital, then you can really limit it, which is why I think SARS-1 was able to burn out. So when we predict what SARS-2 is going to do and when it's going to stop, when will it peak, it can be very challenging to know because the spread it's spreading at different rates than SARS-1. So what we can look at, though, is the numbers from the Chinese. And so they have provided, they're actively providing numbers as we are. And it seems that if everything started in December, they are seeing their peak start to go down right about now. So if you count that time, that's about 3 months. And that's when you start to think about, if you've heard people talk 2 to 3 months. Now, I don't know if that means these economic restrictions are going to go on that long, but that's kind of what you can think of. In terms of, where do we go from here? How do we understand when this thing's going to be over, really is what people want to know. And it can be hard as a scientist to say that, and which is why news people don't really like us all that much, or people who want like hard line answers, because we know it's very nuanced and it's all based on data. And this is a brand new virus and we don't have that data. But what we can think about is some epidemiology. Now, I'm not an epidemiologist, so epidemiologists can comment if they want. But you really are thinking about herd immunity, and a lot of you are familiar with herd immunity. Many of you actually own and operate, herds, cattle herds, swine herds, animals in general, and you need a certain percentage of the population to be immune to something to protect those who are not immune. And 70% seems to be the number that I've been hearing from epidemiologists, is that if 70% of the population is immune, then you could readily then move about and not be as concerned, because those of us who are immune, we're not shedding the virus and spreading it to those who are not immune. And so how do we get there? And there's a couple ways to get there. There's three main ways. The first way to get there, which is quite harsh, and in 2020 unethical really, is that you just let the virus naturally take its course. In that, you would get 70% of the population immune or more, but you would have quite a lot of deaths. And I think with that, you would have an overwhelming influx into hospitals. And that's really, when you think about this virus, what you're trying to avoid. 80 to 83% of the cases of this virus are mild. You will feel crappy. You'll have to self-isolate and self-quarantine, but you'll be okay. You'll get better, and you'll develop immunity. But the problem is our healthcare system is already dealing with the current challenges that people get. Influenza, I mentioned, many more people get infected and die from influenza, and that's already what our healthcare system is dealing with right now. So if that's this baseline. So you're dealing with, if this is the level, you're dealing with influenza, you're dealing with COPD, people with cancer, anything that the healthcare system deals with. And, when I talk to my friends who are medical doctors or students, intensive care units are already operating at a very high percent. So if you add, let's say, a certain percentage of the population, if you just let this thing go ahead without any public health restrictions, you're going to add on top of this a certain amount of people that need immediate care. And this is what Italy is dealing with right now. Italy was not able to control viral spread in its population, and today an additional 624 people died. And that's the highest single death rate of any of the countries. And they have a very large elderly population. And what they're dealing with right now is deciding who gets ventilators and who gets ICU beds and who doesn't. And so when public officials are implementing very strict restrictions on people's movement, this is what they're trying to avoid. They're trying to avoid doctors having to decide who gets beds and who gets ventilators, because our healthcare system is not built in the way it is to absorb the amount of people that would need critical care, on top of all the deaths from influenza and everything else, respiratory issues that people have. So naturally letting something spread that has a transmissibility that SARS-2 does, it can be quite dangerous, especially when there are people walking around who do not have symptoms and are actively shedding the virus. So that's the first way. You let it naturally spread, and a lot of people die because you now are taking ventilators off people who don't even have SARS-2, but they have, let's say, COPD or some other type of lung function. The second way is you build immunity through a vaccine, which is a great way to build immunity. It's the best way to build immunity, and why I get vaccinated and why my children will get vaccinated, and et cetera, why you should get vaccinated, is you can artificially induce herd immunity by giving yourself either a piece of the virus or an attenuated or a less, I want to say less virulent, but less infectious form of the virus. And then you build your own antibodies against the virus. And then you don't get infected, or if you do, it's more mild, you don't shed as much virus, and you're not passing it to people who are immunocompromised or who are elderly or who are children. So the second way is by vaccination. The problem with that, while we do have excellent technology and we can come up with candidate vaccines very quickly, I think you'll have seen a lot of the positive news coming out as all these different companies are coming up with vaccine candidates for SARS-2. There's many different options. The problem is safety trials. You can't just take a vaccine that a company makes and start giving it to the population without doing animal studies, clinical testing, safety trials, and those will be sped up. What the process is in a very intense situation like this is normally you would do animal studies. If they work in animals and it's safe, then you move on to clinical trials, like phase 1 clinical trials, and that's how that progresses. Now, the way this is working, we need a vaccine now, so probably animal testing and safety trials are going to be happening at the same time. You may have seen in the news in Seattle that the first patients were being injected with the first candidate vaccine So they're doing animal trials and small pilot clinical trials at the same time. But if I've been hearing 18 months, if they can develop a vaccine in 18 months, it will be the fastest amount of time that a vaccine has been made in our history. So it would be surprising to me if 18 months was achieved. I would say longer. So we can't rely on vaccines right now to build herd immunity to this virus. And so then that leads me to the third thing. And the third thing is what we are seeing in the United States and many other countries is social isolation, shutting down parts of our economy, and telling people to stay home. And the goal of that is the virus will still spread. It's still here. It's still going to pass asymptomatically between people, but you're going to spread it out over a longer period of time, and you're not going to overwhelm the healthcare system. So I think the main point is not that so many people will die in terms of comparing it maybe to other viruses. It's just more people will die from other things because the healthcare system cannot accommodate that amount of people in a huge amount of time. So that's what we're looking at when you're trying to think about how do we help, stop the spread of this virus until more people are immune. And I think a lot of places you're going with the third option, because the first option is not possible in this type of society. It's not ethical. The second option, we won't have vaccines. So the third option is public health measures. And so that leads to, I get a lot of questions about, I'm just going to look at my phone because I got a message and see what Carrie is saying. Oh, she's so sweet. My 4 o'clock appointment actually got canceled. I was going to have a conference call with somebody about antivirals to this virus. We can talk about antivirals. It's different than a vaccine. It's basically a way to stop the virus from replicating itself in you. So it's not a prophylactic where you can build immunity and prevent against infection, but it's you have the infection, you treat with an antiviral, and then it's going to kill the virus. It's going to neutralize the virus. There are a lot of candidate antivirals. If the United States was able to prepare for this type of pandemic or epidemic in the way that it should, we would have already have screened, determined which antivirals is successful, stockpiled them within our government stockpiles like we have for flu. We have antivirals for flu, which is another reason why flu is a different beast, because we have antivirals for that. They're not, it's not like they're always effective, but we have stockpiles in our government for that. We don't have anything for coronaviruses. But the good news is there are people working on it. There have been a couple, remdesivir is one. It's a nucleoside analog. It basically helps disrupt the replication of the virus. It was used for Ebola. And people say, oh, how is it used for Ebola and used for this virus? Let me see here. Yeah, I will answer Carrie's question in a bit. Lots of viruses have similar symptoms, so just because you had symptoms doesn't mean you had COVID-19. Right. The problem is we don't know. We don't have the testing capacity. It would be hard to know if you had symptoms that were similar to COVID-19 based on the CDC website. You can go on there and see you have a hard time breathing. It's different from flu in a couple different ways. You can read up on that. It would be hard to know because we don't have the test. And I think for a long time, I think you had to show evidence that you had traveled outside the country and that some other restriction to get a test. I don't know if those have relaxed a bit, hopefully as more have been ramped up. So that's a great question. We just don't know. So I'm gonna go realistic answer when a vaccine will be ready, longer than 18 months, unfortunately. But back to antivirals, there are antivirals. Remdesivir, it's by Gilead, which is a pharmaceutical company. That's one. And then there's been a lot of conversation about hydrochloroquine. I have to remember exactly what it's called, but there was a small clinical trial done. Hydrochloroquine can disrupt the ability of the virus to, when it enters the cell, the pH inside the cell little vesicle changes that allows the virus to grab a hold inside your cells. And chloroquinolone, or I don't know how to pronounce it, somebody can correct me later, it disrupts the pH balance and the virus cannot bind inside the cell anymore. So I think it's an anti-malarial drug, but in a small clinical trial. The problem with all these antivirals is it would be difficult to ramp up production in a short amount of time. It's going to take a while. Most of these things take a long time to scale up. And because our reaction to this virus was delayed, it was challenging to build up some of these reserves. So let me go back. Sorry if I skipped. Sometimes I get started with a question and it's just so exciting. I had to keep going. Let me see if I have Carrie's questions. Where did it go? Okay. If we get exposed, are we immune to it going forward? It's a great question. I kind of lean on my coronavirologist mentors that I trained with at Ohio State for some of this background knowledge. But from what I understand, there are coronaviruses that cause just common colds. And from previous evidence of those, you do have antibodies against those, but they're a mild disease. And when you think about some of the more severe diseases, SARS and MERS, some of the studies show that you have antibodies against them and you are protected, but you might not be completely protected. You might still get a mild form, but it's still mild. It means that you're not going to spread it as much. And so it would be the case that people should build immunity to this virus, but how long is it going to last is really the question. But hopefully it would last. It would be enough that you could basically dampen the spread to populations of people like who are immunosuppressed, who have comorbidities that may be susceptible. So those questions are excellent questions, but we're just kind of too early. You're going to start to see antibodies, which is what we say when you become immune. You develop antibodies in your serum. You have serology. That takes a while. It's called the adaptive part of the immune system, and it takes a couple weeks for that to develop. And so you would have to wait after you're done shedding the virus, after your symptoms are over. Somebody would draw your blood and see, okay, do you have antibodies to the virus? Which would be a very interesting and helpful test to have, because soon you could identify a workforce that's immune to SARS-2, and those people could be in hospitals working with patients who have primary SARS-2 so that they are protected and can work with people who have active infections. Is it really no worse than the flu? That's a really hard question. I mean, I am big on telling people that the flu is bad and people don't take it as seriously as they should. There are so many people who die from the flu, and we have a vaccine and we have antivirals. In certain populations, well, I'm pregnant, so we don't have a lot of data on pregnant women and this virus. So I'm going to say not me, but people my age who are not pregnant, it's not going to be worse than the flu. I think what we learned about this virus is its transmissibility, and it seems that the the ability to spread to other people is higher. And just the fact that dealing with this is on top of what we deal with the flu. So who's in the hospital for the flu, we're going to be adding patients to that. And we don't have antivirals, and it's not, the flu is just not, coronavirus isn't something that we've dealt with at this level. And from what I understand from flu virologists, that there's no such really thing as, somebody can correct me if I'm wrong, but I don't think there's this long period of asymptomatic infections where you're shedding influenza, but you feel great, which is what's happening with this new coronavirus. So I think that affects our ability to, okay, who's sick? If you're not sick, but you're shedding, it's a problem. Okay, so those were Carrie's questions. Let's go back to my screen where I'm living. I had to look at my face there. I'm going to put up like a PDF in front of my face so I can just look at your wonderful questions. Let's see. I was just swabbed. Oh, great. Oh, after symptoms started, flu test was negative. She's going to wait for the COVID-19 results. I hope you're negative too. That's interesting. That's great that she was able to get a test. That makes me feel good. There's a question. It's a good question. Is there any truth to the malaria vaccine being used to cure the virus? This is from Dwayne. This probably came from, and before I say this wrong again, let me just look it up. It's quinone. Just don't want to be wrong here. Oh, that's not it. I'll find it. Either way, I think you're just switching up one term, not a malaria vaccine, but it's a malaria antiviral. And the antiviral can work in such a way that it actually inhibits the ability of the coronavirus to replicate inside your cells. So as I'm looking up this drug, I'm going to plug somebody that you all should definitely follow. He's a virologist from Colombia, and he has a whole podcast full of information about viruses called This Week in Virology. I can post this on Carrie's page. He is putting out podcasts at a rapid rate to try to better understand. Okay, it's hydroxychloroquine. I'm sorry, hydroxychloroquine. That's the drug that people have been talking about a lot because a small clinical trial showed that it showed some effectiveness against the virus. But unfortunately, it's not something that can be ready very quickly. You have to ramp up production. We have to do more trials. You can use these drugs through something called compassionate use. So let's say you have a drug that's not FDA approved for SARS coronavirus, but we don't have drugs. If you use compassionate use, then you can use it, but eventually you need the FDA to approve it for insurance purposes, legal purposes. And so actually getting an FDA approved drug that you can scale up production and use rapidly takes a while. They've heard of waves of numbers of people getting sick or people getting reinfected. There are waves of people, I think, getting sick because I think people, as community spread happens and the fact that people are asymptomatic, you're going to have a lot of people getting infected. And if you're in a high risk population, that could be where that comes from. Getting reinfected would be surprising if we have data on that right now because the virus is really just starting to spread in the United States. There were some reports from China that reinfection maybe was happening. And I guess what I could say about that is it's not impossible that you can get reinfected. I think the thing is it would be more mild. You would hope that the immunity would be something called sterilizing immunity, where you completely neutralize the virus and you don't get sick and you don't shed. You don't shed is really the key. But I think that you could get infected and then be reinfected, but you're shedding less. You're not as, it's a more mild virus. Yes, it's interesting. There have been data that shows certain blood types have different people with different blood types have different susceptibilities to this virus. And yeah, that is true. That was a recent study that just came out. And I don't know, I can't remember specifically which one they said, AB, I can't remember, but it does have to do with the ability of viruses to use certain proteins in your body and your immune system to respond to that, which is why that happens. We know that rotavirus, another virus that I work on, it also can matter what blood group you are as to how that infection is gonna proceed inside your body. So that variation in who gets infected and who gets sick can also be blood type specific. Are immunosuppressed people more susceptible no matter age? That is true. Unfortunately, your body's ability to fight the virus is really key. And so no matter what age, if you're immunosuppressed, you should take extra precautions, which can lead to, people who are pregnant are sometimes described as immunosuppressed, but really it's a little bit different in terms of how the immune system works. But they should also just take precautions, particularly in the third trimester when you're more susceptible to influenza infections. So we don't really have the data yet. And so it's best to just err on the side of caution. I hope that, okay, let me see if there's other questions. There's so many things I could talk about, but it's already been like an hour and 15 and I'm wondering, oh yeah, Carrie had asked a great question there about the bovine coronavirus vaccine and if it could protect. And no, it can't. And the reason is because it's that coronavirus, it's a corona, but it's so different from SARS-2 that the antibodies that you would build, if you injected yourself with a bovine vaccine, you would build antibodies to the bovine coronavirus, and bovine coronaviruses aren't going to infect humans, so it would be kind of pointless. And so that's really a distinction between how different those viruses are as to why that's like that. I actually have had a lot of people ask about canceling events, postponing weddings. Should I even go see, my mother or father? Should I, how far should I take the social isolation? And I think that, based on my explanation of the 3 ways that we can stop the spread, let it happen and overwhelm the hospital systems, have a vaccine, or social isolation. And number 3 is key. And so I think if you're trying to predict what policymakers are going to do, if you have a wedding, I was talking to somebody, like, let's say in May, I mean, it's going to depend on if you can financially move it, but I do worry about how long, because 3 months, I'm hearing a lot about 3 months. And so I think you could err on the side of caution if you were wondering whether you should go somewhere with people and you should just try to maybe move that or postpone it. You know, in terms of visiting your family members, you just want to be careful if they're older. You don't want to congregate in groups. I think the name of the game is flatten the curve and try to spread this thing out over time so we don't overwhelm the healthcare system. Okay, let me see if there's anything else. PCR, somebody, let's see. Oh, this is a great question. You mentioned the same subcategories, bovine type. Is there potential to zoonotically jump? So that's really hard to know right now, but people they're going to start studying it. And what we know about coronaviruses is every coronavirus has an animal reservoir, which is different from, let's say, poliovirus or measles, where you could actually potentially eradicate polio because it doesn't hide in any other in any other animal reservoir. It only replicates in humans. And if you get every human to vaccinate, you can eventually eradicate that disease like we did with smallpox. But because coronaviruses hide in animals, we're never going to get rid of coronaviruses. Now, could it jump into bovine? I mean, they're pretty distinct viruses, but as I had described, I erased it. But as I described that, these viruses do have the potential to mutate and adapt to other receptors in other species and maybe grab a hold of that receptor in that animal and then start replicating. So I think it's unlikely. It may be a species. We just have to see, if the virus binds to a receptor in our lungs named X, do other animals have X? And is X as similar as it is in humans? If it's similar, there's a potential there. We also raise animals differently in this country than we do other countries. We don't interact with them in the way that, living in close contact with them as some other countries. I mean, especially in the swine industry and in poultry, we really try to, biosecurity-wise, keep things very contained. And so there's not a lot of interaction with tons of people and these animals. So it would be hard to predict if they're going to jump into those species. But I don't think that's something we have to worry about right now. I think we have other bigger things to think about. But it's something that people at Ohio State and other places, I'm sure, will be looking at, is if you ever came across a domestic animal that contained or was shedding the virus. I think that would be a pretty shocking discovery, and we would have to deal with that as it came. So it's a great question. I don't think it's anything you have to worry about right now. I think there was some talk about, can it infect dogs? Because there was a dog that came up PCR positive. I mean, I think that the dog could have ingested the virus, but I don't think we are seeing dogs replicating this virus. It's not impossible. Again, these viruses can jump species, but I don't think that it's as credible maybe as the news media was reporting. Okay, so I mean, I hopefully answered a lot of questions, and I would be happy to answer anything else you have. You can email me. I'll have Carrie put out my email, but it's stephanie.langel, Langl, @duke.edu. I'd be happy to answer any questions. As this thing evolves, it's going to be really interesting, and I hope people just, I think one of the things that's very hard about what's happening is, yes, people will get sick and potentially, die, but I think the economic consequences, having family members who are going through layoffs and unemployment right now, it's very challenging. And so as somebody who is having family deal with that, but who also understands and why they're doing it, it can be kind of challenging for those two things to kind of reside both in my head. And so it's like, I understand why, because we're trying to flatten the curve, but at the same time, a lot of people are struggling. And when you cancel school is a refuge for a lot of kids. School is a place where a lot of times kids that's where they're getting fed. And in stressful times, there are increase in a lot of domestic issues. And so I don't know. I think a lot about that with this, and I'm glad I'm not a policymaker. I'm glad I'm a scientist who can tell you about the biology of these viruses, but doesn't have to make these type of decisions. But I hope that everyone, can just lean on each other right now, because it's going to be tough for a couple months, but we will get through it. We will build immunity. I mean, that's the good thing is there is an end, and hopefully we will see this virus relegate as its other one, SARS-1 did, either burn itself out or potentially become a mild form like some of its other cousin coronaviruses that just cause colds in humans. So I think with this, I'm going to go, but I'm glad people enjoyed it. And again, if you have other questions, you feel free to ask me, and just stay strong. It will get better. It's going to be rough for the next couple months, but hopefully we will see as we go into summer, things get a little bit better. Okay, well, thank you, Carrie, too, for this opportunity to have me on your page. It was a good time, and I enjoyed it. Okay, bye, everyone.